for the two, Bacillus anthracis and Bacillus mycoides. All of them are motile in nature, right? Then we say they are endospore forming. Then we came to the anthrax. We talked about the anthrax, the disease anthrax. It is like it dates back to a very ancient times, and people they started working. The scientists they started working on that, and this is how the Robert Koch come up with the Koch postulates. Pasha came up with the vaccine. So, what were the alternate names of anthrax? They were like splenic fever, the Chaban. In humans, we can call them as Bull-Sotter's disease, Rack-Picker's disease, malignant carbuncle, and malignant pustule. Then this was the classification, not that much important, right? Then we define, how could we define the anthrax? Anthrax can be peracute, subacute, or acute bacterial infection, which is generally affecting large number of animals, right, from the herbivores to the human beings, right? And it is basically characterized by the septicemic condition and sudden death. That is why it is sometimes it is a peracute, subacute condition or an acute condition, which is basically accompanied. This if the sudden death is accompanied by the exudation of tarry blood from the natural orifices. Then we discussed about the morphology. It's a gram-positive rod, non-motile, having endospore. Then we talked about this Macfadyen reaction. Macfadyen reaction, with which dyes we were using. We discussed that in Macfadyen reaction we were using the polychrome methylene blue dye was the most important dye, which when you use this dye on the clinical sample suspected for the Bacillus anthracis, what do we get? We can see the blue bacilli along with the and surrounding by the pink capsule. Because we know that the Bacillus anthracis, it is a capsulated organism, and this is capsule. Having a capsule provides it an virulence attribute or gives it a virulence characteristic, because of which it moves inside, and because it is antiphagocytic in nature, and it moves inside and cause a disease condition, and such a fatal disease condition it causes. So it is one of the presence of capsule is one of the pathogenic attribute of this Bacillus anthracis. We discussed about the transmission of this thing. Then again we talked about that how could we classify them into different types. Then this was a cycle, anthrax cycle. Moving on, epidemiology we talked about. Coming to the pathogenesis. Pathogenesis is the most interesting this thing of Bacillus anthracis. We said that Bacillus anthracis pathogenesis is mainly related to the plasmids it is harboring within them. Now, plasmids are the extra chromosomal DNA material. Whatever virulence characteristic or whatever pathogenic power they have, it is not because of the genes which is found in the genetic material, but it is because of the genes which are located in the extra chromosomal genetic material, like that is plasmids. So they said that this Bacillus anthracis, they have got two kind of plasmids. They have PXO1 and PXO2 plasmids. Each plasmid encoding certain characteristic, because of which they get this virulence character. PXO1, it is basically responsible for producing certain three type of toxins: the lethal factor, the protective antigen, and the edema factor, and which causes and how they cause the they regulate the host immune system and help in the dissemination of this infection. PXO2 plasmid, it is basically responsible for having the genes which encodes for the capsule production. Capsule is again it's basically it is the, what do we say, it is the structural attribute or the structural characteristics which is a structural entity of this organism, because of which the organism gets a power of antiphagocytosis. That means they cannot be phagocytized by the host immune system, host phagocytic cells, neutrophils, macrophages, and this is very important property. Okay, now for the dissemination or for causing the disease in the animals or in the host, I should say. Now this capsule, what is the major important component of this capsule which is responsible for causing this, for having this antiphagocytic property, is this poly-D-glutamic acid. Many times a question is asked what is the constitution of the capsule of Bacillus anthracis, which provides its the antiphagocytic property. So you should know, you should remember this poly-D-glutamic acid is the major constituent of the capsule present in the Bacillus anthracis, which is responsible for causing the giving them this antiphagocytic property, right? Now moving on to the PXO1, which is another important, which is a plasmid which codes for three important antigenic components, toxic components basically, the protective antigen, the lethal factor, and the edema factor. Now one very important thing to be remembered here is to be emphasized here is these three toxins, they always act in unison, I mean in together. Whenever they are together, like protective antigen along with the lethal factor, it will act, and protective antigen along with the edema factor, it will act. Neither of them is going to act alone, because alone they cannot cause any powerful effect to be pathogenic into to make it a pathogenic this thing, to give it a pathogenic potential, I should say. So this all the three toxins, they have to act in unison or in contact with the other factors, so that, or in along with the other factors, so that in order to produce a virulence ability. So protective antigen, basically this protective antigen is what it is does is, I'll make you understand with the help of a diagram also. This protective antigen is basically the most important factor or the most important component of this toxin factor in the bacillus. Why? Because this is the antigen which is binding onto the host cell. Because as you know, this is any pathogen, as a matter of fact, whether it's a bacteria, whether it's a virus, or whether it is the fungus, they have to come in contact with the host cell in order to cause whatever effect they want to cause on them, right? So coming in contact with the host cell, that is binding onto the host cell, having the receptor for, or recognizing the receptor where they could bind on the host cell, is a very important component of any pathogen. Any pathogen has to come into contact with the cell for causing any pathogenic effect. So here this protective antigen is the most important antigen, which is responsible for binding of this pathogen or binding of this Bacillus anthracis onto the host cell. Now this protective antigen comes in contact with the host cell, it binds onto the host cell, and then it allows the movement of the lethal and the edema factor and that's how it forms a complex. The protective antigen, it forms a complex onto the cell, and that's how it will help in the movement of this lethal and the edema factor inside the cell. And then when they move inside the cell, they produce their own effect, right? So let me share one more thing with you. How a person or how a host cell can get an infection, either by the inhalation, cutaneous route, or by the gastrointestinal route, either by the ingestion, inhalation, or by any abrasion on your skin. You can get these are the spores. The spores, the endospores, they are taken inside into the phagocytic cell, and there they germinate, they move, they in the presence of oxygen and everything, because they are being provided with the very good environment here, they get germination. The spores we are talking about, right? They germinate, and then the bacillus, the vegetative forms start coming out. Because of the once the vegetative forms starts coming out, there is the lysis of this whatever cell it is, macrophagic or whatever phagocytic cell is there. There will be lysis, and all the organism will move into the surroundings. Okay, this is how a vegetative bacilli will come into the surrounding. Once the spore, once an individual is exposed to the spore, what happens, and then the vegetative cells come into the surrounding of the cell. Now, what is happening over here is Vegetative cells may, as I mentioned, now there will be expression of all the important proteins. There will be expression of all the important components which are virulent in nature. As I said, the protective antigen, the PXO1 plasmid, will first give rise to the protective antigen, express the protective antigen, and it is this protective antigen which has got a property of binding onto the host cell. Protective antigen attaches to the receptor, cell target receptor. This antigen, the protective antigen, it will bind onto the receptor, and this protective antigen, it is then it is cleaved by the proteases and it is moved into the different units. It is a single entity right now, right? Protective antigen. Then once it binds onto the host cell, it is cleaved by the proteases, enzymes of your host cell, and it will move into different units, PA20, PA53 and such. And as such, these majority of the units combine together. You can see how these units, they are combining with each other and they are forming the PA, protective antigen receptor complex is formed, which is in a kind of heptamer. Heptamer means actually the 7 units of P protective antigen will combine together and it forms the complex, and this complex attaches onto the host cell, and this is how the complex, you can see it is outside and this portion is inside the cell. Okay, so this is how it is forming probably it is giving rise to the whole kind of condition. A hole is formed on the cell with the help of this protective antigen once it binds onto the receptor of the host cell. And then these are the two factors, the lethal factors and the edema factor. This is one is the lethal factor and this another edema factor. Now, through this hole or through this way which is formed by the protective antigen, these two factors will be endocytized or they will move inside the whole cell, right? These two will move inside the cell, they will be endocytized, and then there, once they move inside the cell, they will start producing their effect. So what is the most important point here is, we are talking about protective antigen. The protective antigen comes and binds onto the receptor of the host cell. It is then cleaved by the host cell proteases enzyme into different units. Basically, the 7 units it will form, and this 7 units, it's known as PA receptor complex, and it is in the form of heptamer because there are 7 units of PA, which is this protective antigen, which is taking part in the formation of this kind of assembly complex. And then through this complex, the lethal factor and the edema factor is going to move inside. Now, what is happening is, these are the factors, like these are the factors which are encoded by the PXO1 plasmid. आया आपका स्पोर से, जिस भी तरीके से आपने स्पोर को अपने बॉडी में लिया, स्पोर will germinate. The vegetative cells, they are going to come out. Vegetative cells, they are going to come out, and then once the vegetative cells come out, the plasmid which is present in this vegetative cell, they start expressing their virulence factor. First of all, they'll start expressing the protective antigen, the lethal factor, and the edema factor. And another is a capsule is also there. So what these lethal factor and protective antigen does is, the सबसे important point is the binding of the cell, binding of the pathogen onto the cell is done with the help of protective antigen. But then this protective antigen allows the movement of the lethal and the edema factor inside the cell. Now here the most important point is, no cell, here you can very well appreciate that if the PA molecule, if the protective antigen molecule is not working, it is not attaching, it is not providing the way for the other factors to move inside the cell, there will be no pathogenesis, right? So what is happening is, PA antigen or the protective antigen is basically the most important antigen in all kind of what do we say, the vaccines which are prepared against anthrax. They basically use the protective antigens. Okay, the protective antigen ke against again antibodies or whatever immunity or you want to raise, if it is raised against the protective antigen, then your body can very well take care of all the infection if you get afterwards, right? So protective antigen, now this protective antigen along with the lethal factor makes, coming to the toxic effect. Now what kind of toxins are produced? Now as I mentioned earlier also, that neither of the factors like lethal factor akele, edema factor akele, they cannot do anything alone or present singly, they cannot do anything. There has to be presence of protective antigen in order to convert these factors into toxic effect. Okay, so protective antigen combines with the lethal factor and it gives rise to lethal toxin. Remember that. Protective antigen combines with the lethal factor and it gives rise to the lethal toxin. Similarly, protective antigens combine with the edema factor and it gives rise to the edema toxin. Now, what are these lethal toxins and edema toxins? Let me come to that time. This thing. Now, what is सबसे पहले हम बात करेंगे edema factor की. Edema factor is basically a cyclase, the adenyl cyclase enzyme. So what it does is it converts the ATP into the ACP. It breaks down the ATP molecule, and it is basically a cyclase enzyme. So it breaks down the ATP molecule and increase in the amount of cyclic AMP will occur. The increased level of the cyclic AMP is seen in the body. Cyclic AMP, this edema factor की बात करना है, ठीक है? Edema factor along with the protective antigen is known as edema toxins. Edema factor, what actually the edema factor does is, it is basically the adenyl cyclase. It acts as an enzyme, right? And it increases the amount of cyclic AMP within the cell. Now, high amount of cyclic AMP within the cell attracts the water from outside the cell, and it disturbs the water homeostasis within the cell. Now, because of this disturbance of the water homeostasis, that is why the name edema means how is your cell swelling up? How is your cell is increasing in the size? Why it is increasing the size? If the increase in the size of a cell, it is basically because of edema thing, right? It is known as an edema. So how it is happening is, this edema factor, it is basically an enzyme, acts like an enzyme, adenyl cyclase enzyme, or cyclase, or calmodulin dependent adenyl cyclase, which increases the amount of ATP inside the cell. Increase amount of ATP because of increase amount of cyclic AMP, I should say, because it breaks down the ATP into cyclic AMP, and increase amount of cyclic AMP within the cell causes more of the water from the outside to move inside the cell. And because of increased amount of water inside the cell, there will be swelling of a cell, right? And this is how the edema factor takes place. This is how the increase in the size takes place. You can very well appreciate the splenomegaly word, like increase amount of the spleen में आपका size of the spleen, it increases to very large extent in some of the ruminant species, and this is because of this edema factor, right? So edema factor, how does it move inside? It will always move inside once the protective antigen is coming. Okay, binds onto the cell. The edema factor अंदर आया. Edema factor, what it does is, it is basically an adenyl cyclase enzyme which causes the increased amount of cyclic AMP, breaking of the ATP into the cyclic AMP. Increased amount of cyclic AMP, water homeostasis is disturbed. Homeostasis between the cell and within the cell and outside the cell, the amount of water which should be there, it is all disturbed. Now, once it is disturbed, more amount of water is taken inside the cell, and because of it, all electrolytes and whatever is there, more amount of it is taken inside the cell, because of which causes the increase in the size of the cell, and that is what it is known as edema condition, right? Excessive fluid accumulation will be seen inside the cell. Now, this along with this thing, what is edema factor or the edema toxin does is, edema toxin has a very good effect, or it's the principal effect or the principal target जो होस्ट सेल का होता है एडिमा फैक्टर के लिए, न्यूट्रोफिल्स होते हैं. एक इफेक्ट तो हुआ कि वाटर होमोस्टेसिस को उन्होंने किसी तरह बिगाड़ दिया. वाटर होमोस्टेसिस is happening is increase in the amount of cell, the water electrolytes inside the cell, and edema is taking place. Now, this edema factor, another target it will act on is the neutrophils. Neutrophils are the another target organism, or the target, not organism, I should say, another target, primary target cells of host which the edema factor targets, and they will try and destroy the neutrophils. And neutrophils, as you know, they are the primary defense mechanism, or they are probably responsible, they are the primary cells in the innate immune system, and they take care of all the pathogens which are moving inside the cell, all the kind of antigens moving inside the cell. They cause them, they phagocytize them, and they take care of them. But this edema factor is smart enough. It will not only cause the whatever it is causing inside the cell, it will also move on to the neutrophil and destroy the neutrophil. They'll inhibit the function of neutrophil, that is phagocytosis. एक तरीके से ये phagocytosis को ये भी वो कर रहा है. दूसरा, हमारा कैप्सूल तो है ही है आपके ऑर्गेनिज्म की सरफेस पे, It has got poly-D-glutamic acid, and because of the capsule also, the phagocytic ability of all the phagocytic cells, you talk not only neutrophils, the macrophages, whatever phagocytic cells are there, the dendritic cells, whatever they are there, they are all not be able to phagocytize this organism because of this capsule. So this is how the anti-phagocytic effect. Now we talked about the edema effect, edema factor. Moving on to the lethal toxin, the lethal factor. Lethal factor, I should say, when lethal factor comes in contact with the protective antigen, it forms the lethal toxin. Now, what does this lethal toxin does? Lethal toxins are basically, from the very word lethal, they are proteases. They act as a protease enzyme, zinc metalloproteases, है ना? And what that does is they cause, because they are proteases, to what all proteases means, the enzyme which will act on different, they will try and causes the lysis of different proteolytic effect. They have a different proteolytic effect on different the cells. So what they do is they cause excessive amount of cytokine production. Different cells को destroy करेंगे lethal toxins, and because of the destruction of different cells, there will be excessive amount of cytokine production. And cytokines, as I earlier mentioned also, I don't know whether you have studied in it in your immunology class or not. Cytokines are the cell, these are the molecules which carry information within the within your immune system from one cell to another cell. So all information is carried within your immune system from with the help of these cytokines. Cytokines, the interleukins, the tumor necrosis factors, these are all cytokines. They are information carrying cells, or the information carrying carriers which move from one cell to another cell and carry information. Now, excessive amount of cytokine production, excessive amount of activation of your cytokines, or excessive amount of activation of your immune system will always be lethal for the host cell. Right? That is not to be done. Okay, everything has to be in their limits. So excessive amount of cytokine activation, excessive amount of your immune system activation will lead to the lethal effect, or will lead to the something, will cause damage to the body, right? So what is here the lethal toxin is doing is, it is a protease, and it moves inside the cell, and it stimulates the macrophages to release the cytokines. Cytokines could be tumor necrosis alpha, TNF alpha. They can be interleukins. Interleukins are there. Interleukin 1, B1, interleukin 2, interleukin 1 beta, whatever. Now these interleukins and they will act on different body organs, different cells of your body, and they started producing necrotic effect, started destroying those body cells. And because of the death of the cells, it is what is termed as necrosis. It will lead to the necrosis. It will lead to the darkening of the tissue. It is all because of these simultaneous effect which is taking place. Now there will be septicemic effect. Now what is septicemia? Septicemia is throughout the body, when the toxins are released by your cells, or toxins are released into the cells, and it moves in your vascular system, and it moves throughout the body with the help of vascular system, with the help of your lymphatic system, then it is termed as the septicemic condition. Now this septicemic conditions start occurring, and because of this septicemic condition, there will be IVF, in increased vascular permeability, because all the cytokines, they are how the messages are carried, it is all in the vascular system, the messages are carried, right? So septicemic conditions हो रही हैं, cytokines release हो रहे हैं, there will be increased vascular permeability. Increased vascular permeability होगी तो obviously आपके red blood cells आपके जहाँ पे उनको होना चाहिए blood vessels में, वहां से move करके extravascular tissues में आएंगे. There will be hemorrhages will be there, right? So this is all how it is correlated, right? It is all related because of increased vascular permeability and because of excessive hemorrhages, blood loss होगा आपकी body में. And this is one increased vascular permeability के कारण you can see the effect very well after the death of an animal, or because the vascular permeability is increased to an extent that the blood, it will start oozing out from all your natural orifices, from the nose, from the ears, from the mouth, from the anus, from everywhere the blood is going to ooze out. This is all because of increased vascular permeability, and it is all because it had happened because of the septicemic condition, and it is all because of the toxins which were released, and it is all because of how the edema factor, the lethal factor, they acted together in unison with the protective antigen, and in relation with the protective antigen, and as a result, what happened is this all circumstances or this all event took place and resulting into septicemic and finally the death of the animal. So I hope you understood all these things. Here, the cleavage of MAPKK means mitogen associated protein kinases. These are all the proteins which are present on the different cells which are being affected or which are being targeted by the lethal toxins because it is a protease in nature. Now remember, many times they ask you question, what is the edema factor? Edema factor is basically the uska chemical nature kya hai? Edema factor is basically the adenyl cyclase, right? Calmodulin dependent adenyl cyclase, and lethal factor is basically what it is. It is nothing but it is a proteases. It acts as a protease. Okay. So this the chemical nature of your edema factor and the chemical nature of the lethal factor is very important to be known. So coming to the zest of this thing, what happens here is the protect हमारे if you want to talk about the pathogenesis कैसे होती है? Two important plasmids are there, PXO1 and PXO2. PXO1 plasmid is responsible for giving rise to three factors: the edema factor, the lethal factor, and the protective antigen. I'm so sorry, I should have mentioned the protective antigen first. The protective antigen, the edema factor, and the lethal factor. And second is the we talked about the PXO2 plasmid, and that is the because it encodes for the and it encodes for the capsule, which gives the antiphagocytic ability, right? So here we come to this thing that we got three toxins. We got the capsule ka pata hai, antiphagocytic, that's done. Now moving on to the further what the protective antigen does. Now neither of these three toxins or neither of these three factors alone can cause any kind of infection. They have to work together in order to produce the disease condition. Now how they are going to work together? The protective antigen along with the lethal factor give rise to lethal toxin. The protective antigen along with the edema factor give rise to the edema toxin. Remember that, right? And what is the most important thing is here is neither the lethal factor nor the edema factor can move inside the cell unless the protective antigen binds onto the cell. The binding onto the receptor, cell target receptor, is very important. Your protective antigen has to bind on the cell target. Once it binds, it is broken down into different units like PA20 and PA53, and such 7 units combine together and form a structure, a complex which allows the movement of the lethal factor and edema factor inside the cell. And inside the cell, once they move, they convert into toxins and they cause their effect. The edema factor, we know it is an adenyl cyclase, causes converting of the ATP into cyclic AMP, excessive amount of cyclic AMP inside the cell. What will it lead? There is a disturbance in the homeostasis, the water homeostasis disturbance. As a result, influx of large amount of water inside the cell, the swelling of a cell will be there, right? It is all because of edema factor. Another effect of edema factor is it acts on the neutrophils and it causes the destruction of neutrophils. So again, the another mechanism by which the phagocytosis was occurring, the another defense mechanism of your body or your host is being destroyed by this edema factor. Moving on to the lethal factor. Lethal factor is converted into lethal toxins once it moves inside the cell, and this lethal toxin again what it does is the basic chemical nature is proteases. So any kind of protein, any kind of protein entity it finds, it basically acts on it and it causes the destruction of this. So cell mein to there are numerous protein entity. Majority of them, what it does it acts on the MAPK, mitogen associated protein kinases. Now, these are very important factors or very important thing within the cell which helps in the translation and transcription and translation process and making of more and more protein within a cell. So when these proteins are targeted by these lethal factor, which is basically a proteases, an enzyme which acts on the protein and cause the lysis of the protein entity. So what will happen is there will be various lethal effect. It will act on macrophages. This is again going to act on different cells and causes excessive release of cytokine storm, excessive release of cytokines, excessive release of the communication molecules, the messenger molecules in the immune system, and excessive release of anything. Activation will lead to the cytokine storm and will lead to the some kind of consequences, the lethal consequences on the host cell, right? We said that there will be septicemia because the toxins will move throughout the body. there will be septicemia, there will be increased vascular permeability, which is basically leads to the hemorrhages, and this hemorrhages and the movement and the excessive amount of blood coming out from the natural orifices is because of that result, increased vascular permeability and all, right? So further, because of excessive hemorrhage hoga to obviously shock hoga, फिर death हो जाएगा. So do you understand what is happening here?